Welcome everyone. My name is Christina Iptoma, and I am mom to Osteo Angel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we're talking with Dr. Kelly Bailey, and she's going to be discussing her recently published paper, Ewing Sarcoma and Osteosarcoma have distinct immune signatures and intercellular communication networks. Thank you so much, Dr. Bailey, for joining us on OsteoBites today. We're thrilled to have you, and thank you also to our panelist, Vicki, for joining us. Vicki is an osteo warrior, and she's also our Junior Advisory Board President this year. A little bit more about our guest today. Dr. Kelly Bailey is a pediatric oncologist and assistant professor of pediatrics at the University of Pittsburgh School of Medicine. She received her medical degree and PhD from West Virginia University and completed her residency in pediatrics and fellowship in pediatric hematology oncology at the University of Michigan School of Medicine. Her clinical and research interests focus on pediatric sarcomas, specifically Ewing sarcoma, and her lab is focused on studying the intersection of DNA damage and the anti-tumor immune response. The lab is interested in understanding the behavior of tumor immune interactions in Ewing tumors that harbor additional germline defects in DNA damage repair genes. And the translational goal of the lab's work is to determine logical DNA damage immunotherapy combinations for the treatment of patients with metastatic and relapsed Ewing sarcoma. And Dr. Bailey is also active in the Children's Oncology Group Bone Tumor Committee and the new Agents for Ewing Sarcoma Task Force. Welcome, Dr. Bailey, and welcome to everyone joining us today. This paper was actually published in November of last year, and I've been so looking forward to hearing you talk about it. So really excited for today. And everyone, please feel free to add any questions you have for Dr. Bailey to the Q&A. And we'll make sure we get those questions answered for you. And then before we get started, we have a few announcements because we have so many exciting things going on. Registration is now open for our Factor Conference, which is June 22nd to 24th in Atlanta. Early bird registration ends on March 15th, so register now to take advantage of that discount. And what we always say about Factor is that you will come to collaborate and you will leave inspired. It's so good. So please come. We also have a limited number of Factor Travel Awards that are sponsored by our MIB Agents Family Fund. And special consideration will be given to young investigators and attendees from outside the U.S. And applications for that are due April 7th. I'm going to drop both the reg link and the Travel Award application link in the chat. And then season 2 of our popular Osteo AYA podcast launches next week. Osteo Warriors Camille and Mia spill the tea on all things osteo. And the first episode of season 2 will drop next Thursday, March 16th. And they're actually talking to 2 other Osteo Warriors, Daniel and Kara, about what it's like navigating life and campus with limb salvage surgery and amputation. And they discuss why they chose their different procedures and how they manage getting around school. And they also share some challenges and horror stories that they've experienced. And I've gotten a preview, and it's a little cringy, but you're going to totally laugh out loud because they are incredibly hilarious. And then I'm saving the most fun news for last, which is you can still participate in our Outbarking Osteosarcoma fundraiser. Dogs are 10 to 50 times more likely to get osteosarcoma than humans. So any canine and human research really benefit both species. You can purchase a cute Outbarking osteosarcoma bandana for your dog. And then you can also post a picture of them and get your friends and family to vote for your doggy. And we have an amazing panel of judges. They decided instead of peer review, they're doing like dog review. And so they're going to help us pick who wore it best. And so I'll drop a link in the chat as well for that. And then I'd like to thank the sponsor of our episode, the Osteosarcoma Institute, OSI. They are a nonprofit organization led by osteosarcoma experts from top U.S. cancer centers who together are concentrating on the cure for osteosarcoma. The mission of the OSI is to dramatically increase treatment options and survival rates in osteosarcoma patients through identifying and funding the most promising and breakthrough osteosarcoma clinical trials and science. And in addition to advancing research, OSI also provides a free resource called OSI Connect for osteosarcoma patients. Their osteosarcoma experts can discuss available treatments, possible side effects, and provide helpful advice for getting the most out of your visits with your treating physician. and this resource is available in English and Spanish and aims to help patients and families find answers to their questions. So thanks so much, OSI, for supporting OsteoBytes. And Vicky, do you want to go ahead and introduce yourself?
Hi, everyone. My name is Vicky Foy, and I am the president of the Junior Advisory Board this year and finished treatment in May. I'm a first-year finance student at Villanova University right now, and I'm so excited for you to be joining us today, Dr. Bailey.
Okay, so well, first off, thank you so much for the invitation. I'm honored to give it, be given the opportunity to speak today. I'm going to be talking, as was mentioned, about our work looking at Ewing and osteoimmunobiology, and especially being more of a Ewing sarcoma basic science researcher, why we decided to look at both and how we decided to kind of leverage similarities between these two cancers in order to really draw out differences in immunobiology. I have nothing to disclose. So I wanted to start first by just giving a little bit more about what we study in my lab day to day. So we study Ewing sarcoma primarily. We study the intersection of DNA damage, whether that's something that's, inherently wrong with the DNA damage repair machinery of the tumor, chemotherapy that's delivering DNA damage, radiation therapy, and then how the immune system, while all of that is going on in the tumor, interacts with the tumor differently. And, historically, there hadn't been a great way to look at that intersection in Ewing sarcoma in a mouse model. So we've taken on an approach of developing a humanized mouse model of Ewing sarcoma so we can really look at human immune cells and human tumor cells in this cancer and the intersection between the two. And a lot of this started emerging, kind of this story and the immunobiology story we were coming up with at the same time. And it's been very helpful to be able to get some of the really detailed human immunobiology data from the Ewing and osteotumors, which I'll get into in a moment, and using it actually to kind of validate this mouse model too, and be able to say, this is where it really hits the mark, and this is where we might need some improvement. So it's been very exciting. And I always like to start too by giving a huge shout out and thanks to the absolutely amazing women in science in my lab. So Alina's my lab manager, Carolina is our technician, and Dr. Jessie Daly is an absolute rising star in bone sarcoma, biology in the world. So I'm very, very grateful for all of them, and you'll see their work kind of woven throughout this story as well. All right, so I just said a lot about Ewing sarcoma, but this is obviously the focus is osteosarcoma today. So, I'm talking here today again to really how we use both of these tumors and looked at both of these tumors in order to really pull out differences in immunobiology. And so that's why I'm here today. I'm really going to try to focus more on the osteosarcoma piece. So if you have any questions, please feel free. I'm happy to kind of chat as we go along and answer questions, whatever everyone would like. All right, so overview-wise, I'm going to be, talking about the clinical disease and just a brief clinical overview because I know we have a kind of a mixed audience. An immunobiology overview, some data looking at the evolution of immune infiltration throughout kind of your disease course, the approach we took to our study and kind of the thought behind it, and again, why we looked at both cancers at the same time. Then I'll get into some of our data looking at our CD8 T cell signatures and our the macrophage signatures. And then I'll give some thoughts about, clinical applications, next steps, just some important thoughts to kind of consider and that we're continuing to, some points we're continuing to work on. So as far as our clinical overview goes, oops, sorry, it's covering up my, the little bar was covering up my slide there for a moment. So as far as the one of the similarities and differences between these two cancers, so, age of diagnosis for both of these cancers. This is a cancers that usually most commonly occur in teenagers. primary site is usually, the bone. There are rare exceptions when other, sites can happen, but, pelvis or femur for Ewing sarcoma and kind of those bony sites around the knee for osteosarcoma are the most common. Metastatic disease-wise, both have a propensity to go to the lung. In the U.S., both of these are rare tumors, with Ewing being a bit more rarer than osteosarcoma. And fusion oncoprotein-wise, Ewing sarcoma is kind of classically, fusion oncoprotein driven, which I promise I won't talk more about EWS-Y1 today. And then osteosarcoma really isn't one of those classic, always driven by fusion oncoprotein. So, and I'm going to come back to these three things I starred here. And this is one reason, we really wanted to use and look at both of these tumors at the same time, is because of the commonalities in these cancers. So in that, when you're thinking about the immune system, a question that reviewers or people listening to lectures or just, other biologists in the lab start asking is, if you compare a Ewing tumor in a teenager to somebody in an adult, well, maybe their immune system is just different because they're older versus younger. And then also the microenvironment too. Is it really fair to look at the, immune infiltration in a bone versus a lung and actually say it's different because of the tumor, when it could just be different because the lung and the bone microenvironments are different? So by having two different cancers in the same organ microenvironments in the same age of patients was really helpful to us to kind of wrap our heads around this a bit more. All right, so as far as immunobiology goes, again, because I realize we have a very broad, diverse group here, I wanted to go through just some real basics of immunobiology just to make sure we're all on the same page. So this schematic on the left is my attempt at making a bone tumor with immune cells in it. And, really immunobiology is looking at immune cells and interactions within a tumor. Immune cells, and thinking about immunobiology, it is one aspect of the tumor microenvironment. Of course, there's a lot of other factors there as well, but it is one, and I would argue an important factor. As far as what immunobiology can kind of include when you're looking at to ask questions about it, it can include just the number of immune cells there. So just, are there a lot? Is there a little bit? Just straight up number, how much is there? Then you can start getting into subtypes. So are there a lot of macrophages versus T cells versus B cells, and kind of, really getting down into subsets of immune cells that are in one tumor versus another, even if they have the same amount. Localization is also really important. So are the immune cells kind of sitting out at the periphery of the tumor, or are they invading, maybe invading is not quite the right word, but localizing into more of specific areas in the tissue? With the case of B cells, are they forming tertiary lymphoid structures? Where are they? How are they kind of sitting around each other or with respect to the tumor? And lastly, their functions. They can be sitting there, they can be revved up and ready to go and kind of fighting against the tumor, or they can be very quiet and just kind of allowing the tumor to grow and do its thing. So I think the function is obviously also incredibly important on top of what cells are there and how many. And I think with all of this and why we study this, so the kind of translational question, the long goal with all of this is, can we really try to understand the immune system and try to help immune cells start attacking the tumor more effectively at some point along the way in therapy? So as far as what we know about the immunobiology of Ewing sarcoma and osteosarcoma, especially when we were first, getting this story and study up and going, Ewing sarcoma, I will say there was much, much less known about it as compared to osteosarcoma. There have been some more advanced studies already in osteosarcoma, which I'll highlight. I can only highlight a few, so I apologize if there are any researchers that are listening if I don't cite your paper, but I had to keep it brief. But classically, though, Ewing sarcoma is described as an immune desert. And I will put a little star there and say perhaps that's not always fair. But at primary diagnosis, it is true there are very few immune cells around. And that's getting at the second point, that the majority of the studies have examined only primary tumors. So tumors before chemotherapy is started and in the bone. And then very rarely have paired patient samples been looked at over time. As far as osteosarcoma, again, there have been more larger studies for osteosarcoma immunobiology. I gave two examples here. So one that started going in and looking at immune infiltrate and kind of defined subsets. So with low, medium, or kind of high immune infiltration and activity. Another paper from 2020 actually was kind of making an osteosarcoma single cell map, if you will, of all of the cells in the tumor, and pulled out the immune cells that they happen to capture and identified TIGIT as a possible single immunotherapeutic target. And I'll get back to a couple of points on some of these studies later. I think a commonality between the two is that, again, rarely do we have paired patient samples over time studied. A lot of that has to do with just access to tissue, what's available, and I think that I will also mention that kind of toward the end of the talk as well. So we have when we're writing grants, we have our big, our big lengthy hypotheses, and I wanted to make this as lay friendly and interactive, as tangible as possible. So basically boiling it all down, our overarching hypothesis was simply that bone sarcoma immunobiology is not static. So what might happen pre-chemo in a bone might be very different than what happens in a lung at the time of relapse. So to keep it simple. So and now I want to get into timing and why timing might matter. So when we're thinking about looking at immunobiology and bone sarcomas, what could be changing? What do we need to think about, and why might things make sense at one point and not at another point in time? So if you think about kind of a generic therapy timeline, so of course you have diagnosis. And so there's usually, to diagnose the tumor, there's usually a sample acquired at that time. So that's an opportunity to go in and look at immunobiology. At the time of local control, most of the time, there's a surgical excision. And that's another point in time where you can go in and have an opportunity to look at immunobiology. At the end of therapy, there's really, hopefully there's no tumor left around, and there's really no window there other than just looking at peripheral blood to be able to get a sense of what the immune system is doing. And then, of course, if a relapse happens to occur, that's another time when another biopsy might occur, and you might be able to go in and have a new window into what immunobiology is happening. Perhaps it's now in the lung versus the bone, and so it's just a kind of a different point in time. So the immune system itself, of course, before you start chemotherapy, you're not, there's not all of that excessive immunosuppression like you have once you do start chemotherapy. But kind of at baseline, though, just the fact that you have a cancer might mean that your immune system might be revved up in a different way than a person that doesn't have cancer. And then, of course, once you start chemotherapy, that tends to, cause a lot of immunosuppression, especially when you're thinking about looking at samples around the time of local control. That's after, a significant amount of chemotherapy usually. And just keeping that in mind when you're thinking about looking at immunobiology and how the data you get at certain time points might be applied toward like a therapeutic thought or application. And then, of course, after the end of therapy, immune recovery starts happening. So your immune system starts to kind of normalize back out again. But that does take some time. And then at the time of a relapse occurs, then you start getting into effects that the new therapy you're starting, or maybe if this is a time when an immunotherapy is being applied, there can be, of course, effects on your systemic immune system if you're getting an immunotherapy or other sort of relapse therapies. And so I think with this, why timing matters and why I spent so much time on this is because if we're examining immunobiology when we're getting samples, say at diagnosis or the time of local control, it's important to keep that immune context in the body kind of in mind, especially if you're using that data to make an argument for actually giving an immune-based treatment down here at the time of relapse, where perhaps because of the microenvironment or the therapy you went through or your immune recovery, that some of that biology has changed at that point. So I will leave that there for now and move on to kind of some of our initial data that we had and why we really wanted to look at Ewing sarcoma and osteosarcoma immunobiology more in a linear way. So I would like to pause for a second and really give hats off to our absolutely fantastic collaborators here at the University of Pittsburgh in the cancer immunology program. So Dr. Sillo, Bruno, and Vignali, absolute rock stars, leaders in the field, and I have absolutely loved collaborating with all of them. So some of the initial data we did and looked at in osteosarcoma, Ewing sarcoma. One example here, so this is a same patient. So on the column on the left, you can see their primary tumor, and this patient happened to relapse twice. So had a biopsy when they relapsed the first time and relapsed the second time. And you can see in the kind of rainbow colors along the bottom here, the different immune cell or just cell markers that we are using here. So looking at B cells, T cells, and then also macrophages. And you can see, as I mentioned before, Ewing sarcoma has that stamp of immune desert, right? So, and that certainly was the case when looking at the primary tumor. Not a lot going on in that sample from that primary bone site. But at the time of relapse 1, you can see that, the immunobiology starts to evolve a little bit. There aren't a ton of cells there, but there's more. And then by the time the second relapse occurs, there's a lot more immune cells there, but not only T cells, also some of those more immunosuppressive cells like macrophages and also FOXP3 cells. So, I think that this was some of the data that kind of set up the rest of this project. And if you look over at osteosarcoma, we did a similar thing to, again, look at that question of bone versus lung versus relapse and just see if you could, get those similar trends. And certainly you do. So I think one of the differences is that most osteosarcomas have some, more immune infiltration in that primary tumor. So you can see here like macrophages. And then, as relapses occur, the immunobiology can really change a lot in these tumors. And whether some of that is the fact that they are now in the lung, whether that some of it is they were, previously exposed to chemotherapy and other agents, it's probably multifactorial, but just the fact that it evolves is what really got us interested.
And Dr. Bailey, start to clarify back on that slide. So the red, the CD68 are the immunosuppressive macrophages.
Yeah. So this here, at least looking at macrophages. So we didn't really get into what they were doing functionally until our studies later, but yeah.
And these for the relapses, are these off therapy, like off chemo?
Oh, I'm so glad you asked that. Yes. So these ones, the ones we're looking at later, we controlled later for how close you were to chemotherapy. These initial ones we did not control for anything like that. We were just looking at whenever you relapse, let's just go look. Our data that I'll show you with all of our single cell data, we did actually, it was either pre-chemotherapy or at least 6 months off of chemotherapy. So giving some of that immune cell time to recover. So that's a great question. Yeah. And then just using, again, Ewing sarcoma as an example, I won't dwell on this too long because I know this, again, it's an osteosarcoma talk, but we went in and got paired patient samples from the COG biorepository to kind of look a little bit more at this, comparing primary tumor to relapse. So doing an H&E, this multiplexed immunohistochemistry or immunofluorescence to look at all these immune markers and quantifying it. And just showing, across multiple patients here, this isn't even all of them, but enough for the slide, certainly, just that, usually in the primary tumor, there is way less immune infiltration and than in the relapses or distant sites. And that this can also change, it also can change what cells are there over time. So I won't dwell on this too much, but just did want to show that we expanded this out to more patients than just 2. So as far as the approach to the single cell part of our study, so the points we really wanted to consider in this and to be thoughtful about were, 1, the immune age. So we were looking for a data set of just normal, healthy, adolescent peripheral blood that had been single cell RNA sequenced that we could use as a reference point. Sometimes it's nice to have that kind of check to see where your baseline should be. But we really couldn't find anything. So that was one thing we really wanted to include in this study, not only for us, but also for kind of a resource for the community. few immune cells analyzed. So sometimes if you get, a big chunk of a tumor and you are analyzing, usually with single cell sequencing, you're analyzing somewhere between 7 and 10,000 cells in a sample. The majority of that, of course, is always going to be tumor cells. There might be some stroma in there, but the immune cells will be in there, but it's going to be the minority of what you've actually sequenced. And we really wanted to make immune cells the focus of this. So we, which I'll get to in a second, we decided to kind of pull out the immune cells so we could sequence as many of those as possible. Loss of immune cells during processing. So sometimes if you viably freeze cells, and then pull them out later and digest them and sequence them, you can lose some immune cells in that processing. And again, we really wanted to get as much as we could as fast as possible. So we did a little bit of a different approach with that as well. And then the microenvironment, of course, again, why we use these two cancers was because they have the same tumor microenvironments. And as you just very wisely mentioned, the time from last chemotherapy. So all of our single cell sequencing was either pre-chemotherapy or at least 6 months off of any chemotherapy. All right. And again, our 3 stars, same age, same primary site, same site of metastasis for that kind of really good baseline and microenvironmental controls throughout this. All right. So first, as far as our approach to this, the consent and I just have to pause here and say that the selflessness and the willingness of families and patients, teenagers, you can only imagine, especially right at the time of diagnosis or relapse when a biopsy sample is happening, to so generously, give to research and want to participate, pay it forward. All of this. I'm so appreciative of all patients and families that are so excited to do this at such a hard time. So thank you. If anyone is out there, thank you all. As far as our sample acquisition, so of course, all clinical priorities are taken care of first. And then for this study, we obtained 1 tube of peripheral blood and then 2 additional tumor cores, of course, when it was safe for the patient to have those 2 additional tumor cores acquired. As far as the preparation that we use, so we, as I kind of mentioned before, we wanted to make sure we kept as many immune cells as possible in this processing. So we did fresh tissue digestion. So we went from OR right to digestion. And we created single cell suspensions or isolated the buffy coat from the peripheral blood. In order to really enrich the immune cells that we were sequencing, we actually sorted the cells. So we stained them for CD45 and pulled out all of the CD45 positive cells so that most of, actually all of what we were getting were immune cells, and we weren't getting, a bunch of tumor cells in there. And we saved tumor samples, of course, but really we wanted to focus on getting as many of the immune cells in that sample out and sequenced as possible. And then we went on to do single cell RNA sequencing. The parallel samples we also did. So we looked at FFPE. So that's basically like paraffin blocks of tissue that pathology has extra of after they're done making the diagnosis. And that was nice because we could get that sample for this sample that we were sequencing. So we could kind of see where the immune cells were as well. And we could also go back and if that patient, say we're doing our single cell sequencing at the time of first relapse, but that patient had had a diagnostic biopsy and a local control sample too, we could go back and pull those two to kind of look at that evolution over time a little better. These are the same kind of cell markers that I had mentioned before, so I won't dwell on those. And then again, the additional samples was we wanted to create a healthy donor single cell peripheral blood pool that we could look at and see what's normal. And then also looking at healthy lung biopsy tissues from patients and H&Es from specimens. So I'll go ahead and jump into the data. So looking at our T cell signatures and our macrophage signatures, and I wanted to make sure that when I'm showing all of these plots with just a bunch of dots all over them, that we're kind of all on the same page. So I wanted to give a super basic introduction to kind of looking at single cell RNA sequencing data. So of course, we acquire that transcriptional profile from each individual cell. Those cells, and then we kind of plot them on this 2D space to make it just to better wrap our head around what we're looking at, so to visually represent how these cells are alike or not alike. And so the dots on these plots represent a cell, And then the colors we layer in to kind of annotate, so to give the clusters or whatever we're calling them a name. And then dots that are closer together are more similar to each other. So of course, all these clustered around here are very similar, and these guys would be different. I just made up this dot. This is actually not data at all. I just as an example. So let's jump into CD8 T cell signatures here. So you can see, I should say at first, why are we looking at the CD8 T cells? Of course, that's a pretty potentially targetable population when thinking about a therapeutic. So that's why we really focus on T cells first. So if you look first across the top here, so these are all of our peripheral blood samples. So you can see that there's different clusters of T cells from patient blood, from patients with Ewing sarcoma or osteosarcoma, and that it's different from healthy donor controls, and that's to be expected. And that the peripheral blood signatures are different from the CD8 T cell signatures that we see within the tumor. So the tumor infiltrating lymphocytes, that's what TIL stands for. And so you can see that these clusters are different. So what actually are some of these clusters of interest? Oops, sorry. So in the Ewing sarcoma samples, we saw what we call cluster 4 here in the orange. So this is an effector T cell population. So it's revved up, it's ready to go, it's doing its thing, it's not quiet. So that was something that we found uniquely in Ewing sarcoma and not in osteosarcoma. Second, this population in blue here, this cluster 1 that we see in Ewing sarcoma and not osteosarcoma. So these are CD8 T cells that are expressing galectin, which can actually cause an immunosuppressive effect on CD8 T cells. So it suggests that there's kind of a unique a unique immunosuppressive subset of cells here in these Ewing tumors. And then lastly, in cluster 3 here, this is a population that we really saw stand out in osteosarcoma and really didn't see a lot of it at all in Ewing sarcoma. And this is a cluster of terminal exhaustion. So kind of the classic CD8 T cells that are targeted by those checkpoint inhibitors you're always hearing about in the clinic and on TV and all these things. So that's the population there where this kind of classic immune checkpoint exhausted population exists. So just to give a really brief, overview then of what we saw in Ewing sarcoma. So this kind of immunosuppressive galectin-3 expressing subset of CD8s, and then also a population of effector T cells. And then osteosarcomas, so that cluster of the exhausted cells, cluster 3 there, really expressed a lot of co-inhibitory receptors. So not just TIGIT, which I will remember that paper earlier that said, TIGIT's a target. It's definitely there, but a lot of other stuff is there too. And I just say that because perhaps just blocking one of these isn't enough. There's a redundant mechanism here that can kind of keep these cells quiet. And another thing that we thought was very interesting was that CTLA4 was not expressed. And so, sometimes there have been some studies where CTLA4 inhibition hasn't been all that efficacious. So perhaps in some patients this could be why, but this is a very small study, so we can't make a blanket statement certainly. All right, so then moving on to monocytes and macrophages. And again, if you look at the top here, this is a little bit different. So I'm going to show you this group of these dots and then a kind of another subset of them. So first, we didn't just look at one type of macrophage here like we did with the CD8 T cells. We kind of looked across monocytes and macrophages. So this first group of maps here is just kind of looking at these canonical cell types. So in the peripheral blood, so your CD14 positive, 16 negative, your monocyte populations, basically. And then when you move into the tumor, of course, the resident tissue macrophages and the different populations that are there. These don't look strikingly different in their shapes too much when you just kind of break it out by cell types. But then when you go in and try to really start looking at the signatures of these cell types and pulling out other clusters, that's where it gets a little bit more interesting. So I will flip to this next page. And just to orient you, that slide that I just showed with the cell types is up here on the left. And then we break this down farther and to kind of looking at subgroups in here. And so if you go and look at the tumors, you can see that this big blue population here of macrophages, it's now broken into actually 3 kind of subtypes of CD14/16 positive cells. So there's kind of this cluster 2, 4, and 8 here. In the Ewing tumor microenvironment, we saw that there was much more of cluster 2 up here. And then in osteosarcoma, this brown cluster 8 was more extended and there was more of it. And that's a cluster that was it had much greater HLA expression and suggested that there was a lot of antigen presentation potentially happening in this cluster. And so the brief summary for the Ewing tumor piece. So there are, relatively fewer macrophages in Ewing tumors as compared to osteosarcoma, which I know I didn't show pictures for the sake of time, and the macrophages that are in Ewing tumors have a really reduced, if almost absent, CXCL10 and CXCL12 signaling. So that's kind of a signaling mechanism that screams out like, hey, immune cells, come in here, come in the tumor. And Ewing tumors really don't have that signature. And that's very different from osteosarcoma, where there are generally more macrophages. You have a stronger kind of call and kind of inflammatory signal there. And, there's that enhanced cluster 8. So maybe some of these macrophages are doing a little bit more antigen presentation. So I will, for time and in order to allow for questions and discussion, I thought I would start to go through some thoughts and some wrap up with, what this means and where we're going with some of this, at least in the Ewing sarcoma part. So some therapeutic thoughts that I think, single agents are unlikely to be the answer. I think that's true with single agent chemotherapy, immunotherapy, any of it. So, working toward combinations, I think, is really where we need to go with all of this, and understanding when we can partner things together and at what point in time does that make sense. Pairing agents with the current immune status as much as possible, I think, could be very important. So again, if you have data from a pre-chemo primary tumor that you might be trying to apply to something during relapse, that might not make a whole lot of sense. We just don't know. So we need to go in and look and try to pair it up and make sure that signal is still there that we're trying to go after. For Ewing sarcoma, looking at galectin-3 and its immunosuppressive potential and the lack of CXCL10 in the tumor microenvironment are ongoing projects that we're working on. As far as osteosarcoma, like I mentioned before, really thinking about inhibiting multiple inhibitory receptors, or turning off that inhibition, but then also stimulating at the same time is another way you could kind of start to rev up some of the immune cells in osteotumors. Again, potentially, I just gave an example of like ICOS is a receptor where if you activate it, then you really start driving the cells to function better again. And then one other thing, we kind of postulated about in the discussion of this paper was trying to examine or figuring out if we could harness some sort of engineered macrophage specifically for osteosarcoma, because, there's more macrophages there. They seem to kind of like being in the tumor. And, if we could engineer them to kind of work with us instead of against us when we're trying to treat these cancers, that might be a new therapeutic, avenue of interest. We are certainly not working on that, but it's just a thought that we had. And then the other thing that I think is always important too is that, we've shown Ewing sarcoma and osteosarcoma as each as one different, one group and the other group. But we know within each of these tumors, there are subsets of tumors, and probably the immunobiology of some tumor subsets might be very different. So different expression, profiles or groups or mutations that the tumors could have. So I think that's another thing that's important to study, but it's challenging because when you have rare tumors and then you're trying to look at tumor subsets, those numbers get small very fast. So, but I think it should be certainly a goal over time. As far as material, so through my interaction with the Children's Oncology group, we are always very invested and very interested and have been working toward doing this diagnostic biopsy protocol for Ewing sarcoma and osteosarcoma. And I think that immunobiology is another great example of why we need biopsy material from multiple points and having them stored in different ways. Every different storage mechanism, you can get different information out of those samples and use different applications to answer questions. So just to continue to think over time about how we can really optimize better understanding these tumors at multiple points. And I think, getting samples and looking at biology is super important in that. So. And then lastly, if there are any researchers on here, I just really wanted to emphasize that all of our single-cell RNA-seq data from this study is publicly available. So it's on the Gene Expression Omnibus. So I think 2 points I'd like to highlight here as far as, being able to take this data and leverage it with your own work. One, we put our healthy adolescent single-cell peripheral blood controls there. So if you need that reference point for anything, it's there and publicly available. And then also all of our peripheral blood and paired tumor samples from our patients are there. And I think that can be a really useful resource because you can use the single cell signatures to kind of deconvolute bulk RNA-seq data. And that's a really nice way of kind of better refining some of those immune populations when you haven't done single cell RNA sequencing. So that is all out there and shared and go for it. If you have any questions, feel free to reach out. I would once again like to thank patients and their families for your generosity with signing up for not only clinical trials, but biology studies and everything in between. So I'd like to thank the COG Bone Tumor Committee because as I said, I'm a Ewing biologist at heart. So I love the conversations about osteosarcoma that I get to listen to through COG on the biology and on the clinical side. Our division and our Pittsburgh Sarcoma Research Collaborative, my lab that I already mentioned, and the Vignali and Bruno labs, and then my funding. So I will pause there for any questions. And thank you all so much again for the invitation.
Thanks so much. That was so interesting. And thank you also for so nicely kind of helping us decipher those plots with all the.
Oh, sure. Absolutely. Yeah.
And everyone, please feel free to add any questions you have to the Q&A, but I might just kick off with a couple. And also just want to say, thanks so much for sharing your data. And so great that you have that kind of the control, the healthy samples.
Yeah, definitely.
As a reference point. That's really great.
Yeah, it's really important. And, it's a thing where it's it's never the full focus of the study, but it can be really important if you want to use it for other studies and things. So, yeah.
Yeah. And also impressed that you do have those samples from diagnosis, kind of throughout the timeline, because that I know can be also challenging to get.
Absolutely.
But so actually a couple of, I think all the points that you mentioned regarding timing are really interesting. And so I have a couple questions about that, just because
Sure.
Make me kind of realize that's true. All the prior treatment does really affect immune response, the chemotherapy, radiation therapy.
Yeah.
And so I'm just kind of curious, what implications might be for the timing also just of immunotherapy. Because I know right now when we're studying it, it always does come after heavily kind of pre-treated.
Yes. Yeah.
That's right. So which all messes with your immune system. And then, also just thinking about the fact that immunotherapy, and I don't know if this is what your thoughts on this are, but that it can be potentially more effective with minimal disease versus
absolutely. Yeah.
So in terms of timing that, well within the context of, what the other therapies do to your immune system, but also knowing that immunotherapy potentially is more effective in a setting with minimal disease, low disease burden.
Yeah.
So on in the future, when we think about incorporating immunotherapy, where does that happen?
Yeah. And I think there's so much still, left to study. I mean, we don't fully know, all of the immunobiology effects of all of our treatments in the tumor because we can only go in and look in the tumor in very specific times. And, at the time of local control, that's a time, of course, we're hoping that 90+% of the tumor is dead, So I think that, I agree with you that using immunotherapy only to control both big disease is not the way to go. And so thinking about how, when we're in kind of a mop-up mode, if you will, after radiation or after a surgical resection or, sometime when your immune system is good enough to be able to kind of still respond. But you don't have like this massive disease burden that you need to get under control. That's exactly right. So, I think we've been very interested in Ewing sarcoma and looking at immunobiology around the time of radiation for that exact kind of same reason, getting it kind of toward the end of therapy for metastatic disease and using other stuff as a mop-up after. And so I think we're going to continue to study this and to really figure out, exactly what our therapies are doing to the immunobiology and when it actually makes sense to step in, because timing absolutely matters. And even in adult cancers, even the timing of when you start an immunotherapy versus another one could be very important, and papers have shown that. So I think there's still a lot to learn, and I think we're excited to dive in and kind of look at it more. So.
Yeah, I'm curious. So in osteosarcoma, there is the mifurtamide, the MTPPE, is basically an immunotherapy, right? That you could. I mean, they also want you to get it while you're getting chemo, because usually you give it with the adjuvant chemo post resection. And I'm just curious. That might have been an exclusion criteria for samples, but I'm just curious, and what that would look like because that's kind of an interesting thing where you're actually having.
Yeah, I mean that's a hard. That's again, I think that's why some of this we need to back into. I think into animal models, because we don't. If you start a therapy, you're usually not most of the time we don't get biopsies while you're on therapy. We don't know. It's just a black window. Like we might have a biopsy when a relapse happens. Then it's a black box. And then maybe if another relapse happens, we get another peek, but we never really have the opportunity to see what's happening in between. So I think that's where some animal modeling, whether in like mice for Ewing sarcoma or like, dog trials for osteosarcoma, there might be potentially other opportunities for samples or to take a look at, what's going on during therapy. So that's a huge black box that I think it's really important, but I certainly don't have the solution as to how to get in there, at least in the human setting. So, there maybe in the future, maybe we could have some more trials where they're like on therapy biopsies, but I know that's really hard, for patients and families to go through. So, it's always the risk benefit of knowing more versus, having to undergo another procedure and that pain of doing that just for a trial, like they will do sometimes on adult trials for other different cancers. So,
yeah.
We just had a question come in from the audience. They said it was about PNBCs from controls. They said, were these controls matched with the patients, as in their age and their gender matched?
Oh, that's a great question. So the control, so that the healthy, or we don't know if we are assuming they're healthy, but they're blood donors. They're between the ages of 16 and 18. So kind of that adolescent, you can start donating blood when you're 16. So that kind of adolescent age range. And we had both male and female there. So we didn't do one control for every other sample that we had as far as patient samples, but we did have just an adolescent male and female population of just normal peripheral blood. So I hope that answered the question. And, most of our patients were, somewhere between, I think I'd have to go back and look exactly, I think somewhere between like 14 and 19 years old. So that same range is where our control blood was from. Yes.
Another question. I'm reading this question. I'm not even sure what all the words mean, but I think you do. They wrote, do you see a role in creating standardization of longitudinal biopsies across diagnoses, treatment, relapse to improve tumor biology monitoring? And how do you think we can implement this standardization?
I would love any thoughts. No, I think what we can say, is as a biologist or as for COG wise, as a committee or whatever, what would be what we think is the ideal pie in the sky dream for what we would like to in order to really optimize clinical diagnosis, clinical trial enrollment biology, right? So kind of along that whole spectrum. I don't think that it's probably nearly impossible to completely standardize that. There's so many different hospitals approaches, even different surgical approaches for things. But just, I think, getting the word out there and spreading the word more that this is important. There's actually, reasons for it. This sort of thing, I think, can be helpful. And then I'm hoping over time that, people that are doing less invasive tumor monitoring, so like ctDNA, other techniques like that, that can also be integrated, I think, over time into this and give us potentially less invasive window at looking at what the tumor is or isn't doing when we don't have to go in and surgically get a biopsy piece there. So I would love to get as many samples as possible, but I think that again, patients have to undergo all those biopsies too. But the ones we are getting already, I think ensuring that they're optimally processed and stored and made available, annotated. What was this patient? What did they have? That sort of thing is really important and certainly a priority.
Great. We have another question that came in. Can you comment on the importance or lack of importance regarding the spatial landscape of the tumor immune microenvironment?
The spatial landscape, I'm guessing, meaning like where the immune cells are, is I guess what they're getting at. Probably, I'll take that to be what it means. Yes. So I think that it's interesting because certainly the size of some of these tumors, you start anytime you get really, really large tumor like primary tumors, the immune cells do have a harder time getting throughout that whole specimen. But I think when you have like lung metastases, you certainly see more immune cells kind of getting into the whole of the tumor in those situations versus sitting more out at the periphery. But I really think, some of it's probably certainly like cytokine and biology, but I think some of it is frankly just the size. So I think it's once you start cutting off blood vessel supply to some of these bigger tumors and you get necrotic stuff in the center, it's harder to get immune cells distributed through that. So I think it is important. I think that, trying to get the tissue as penetrated as possible with immune cells, especially if you're trying to get an immunotherapy, going, is important. I don't think that we know enough to know how much is enough. And I think it would take a long time to know that for sure, like how many immune cells and how kind of deep in do you need them to be effective. So in this situation for bone tumors, at least. So.
Another was, did you look at the variability of immune clusters in like different spots of the same tumor? Because I know sometimes there's like gene variability within the same tumor.
Yeah, so that's a really good question. So for the single cell sequencing, so we couldn't do that spatial localization with that because we made single cell suspensions of the whole sample that we got. So we lost our ability to see where the immune cells were kind of localized. We just kind of pulled them all out and then did the sequencing. But the paraffin sections that we had of the same tumor, so it wasn't exactly the same core, but it was right, kind of the right next door to where we had our sample from. We could get a better sense there of if these cells were all clustered up in one area versus spread out more, but we don't have the transcriptomic information with that too. So we could know if there were what types of cells were where, but not what their transcriptional profiles were in that localization piece just yet.
Another was there a standardized location within the tumor that you generally used for which the tumor samples were obtained for the single cell analysis? Like, was it the central portion, peripheral portions?
Yeah, so we for better or for worse, we really couldn't dictate where, these were all being done for clinical diagnostic purposes, and then we just got extra cores when we could. So that kind of planning on where in the tumor it was biopsied, especially for the primary bone tumors, we had no control over that. That was just based on safety of the approach per the IR or the surgeon. The lung mets, they sometimes the samples weren't cores, they were just actually like the whole met would come out and we would just get pieces of it. So in that situation, we pretty much got the whole of it. And so in that situation, I think we had a little bit better control over that question of like where in the tumor, you are. I think the mets are easier to kind of get a better representation because they're usually smaller and you can get more of it out in one shot versus just a core at diagnosis. So.
Great. If you have any more questions, keep them coming. We still have Dr. Bailey for a couple of minutes. But a couple other things I just wanted to throw out there too were, so I'm talking about kind of, again, going back to kind of other therapy. So chemo and radiation, there's some DNA damage happening, but there's also, and I think this is being studied, like potential, like a triggering immune response from either radiation therapy or something like cryoablation. So curious if you have any thoughts on that too, or if that was something that you saw in your samples. I don't know if you had samples that had radiation therapy prior to pulling that sample, or
Yeah. So no. So none of them had been exposed to radiation therapy. But we were very interested in that. Actually, Dr. Daly, the instructor that's in our lab, that's one of her main focuses, is really looking at exactly what you said. So radiation kind of causes this burst of DNA damage. Immune cells go, whoa, kind of rush in, and quickly though are kind of quieted back down again. So an immunosuppressive effect to quiet that down again. So she's very interested in better understanding, at least for Ewing sarcoma, like what causes that quieting, and can we delay it a little bit or improve upon, kind of keep it stimulated for a while longer to help clear out that tumor a little bit better. So it's a great question.
So really interesting, and kind of looking forward, and kind of what are your next steps, I guess, coming out of this that you're focused on now?
Yeah, so I think we're taking a lot of our it's Ewing focus, sorry.
That's fine.
But if there's any osteosarcoma collaborators out there, happy to chat. But we're really using a lot of these data again to like look at our humanized mouse model and figure out how close, or if we see a lung met in a mouse with human immune cells and human tumor cells, how close is that to the immunobiology of a relapse in a patient. And so we can use this single cell data to kind of see how close our models are, but then also really going after those couple of inhibitory factors that I was mentioning. So like galectin, and trying to rev things back up in the tumor again is really our focus. So I think that's kind of where we're at right now. So
just one other question I actually want to throw out there, because you were talking about the checkpoint inhibitors and and potentially combo. Because I know there's been like trials with PD-1 and also the CTLA-4, which is so interesting that you said that's not present in osteo, because I don't know.
At least in the samples we analyzed, yeah.
At least in the samples we analyzed. I will asterisk that.
Yeah.
But just thoughts too on, so, there's a combination of checkpoint inhibitors, but and in my very basic rudimentary understanding of how the cell works, I always think of there's the, taking the brakes off, right?
Yeah,
kind of like checkpoint inhibitors. But then there's also just like adding, like stepping on the pedal, which might be infusing with, T cells and NK cells and stuff like that. And so kind of in this, with these different tool sets available, it's kind of, you want to be doing both, like take off the brakes and then also put some gas on something else. And
I'm a huge proponent, as I think a lot of us are in the field, that like probably a single method isn't going to be, the answer. It might be a part of the answer, but not the whole answer. So figuring out, if we throw on drug A, say I'm just making it, say it's a checkpoint inhibitor, what happens then? And if we do think of something like hit the accelerator while we're hitting the brake, does that help? And I think by really starting to again like look at this over time more and try to figure out, like for each patient, like did this drug fail because you didn't express this receptor, or did this did you need that extra kind of hit to really start making headway in the tumor? I think the other thing you bring up there, it was really good, is the idea that, delivering cell therapies and then using drugs to just enhance their effect is a really great thought too. I certainly don't have all the answers as far as like which one is the top priority. All I can do is, I think, continue to be an advocate for continuing to look and not just to treat, of course, these tumors as one thing. Like, each patient's tumor at each relapse or at each metastases or whatever is probably a little bit different, and understanding that better, I think, hopefully will provide better answers and ideas. So
thank you. Well, we appreciate all your work on Ewing's and osteosarcoma. So thank you so much. And thank you
absolutely.
for joining us on Osteobites today and making it better for pediatric sarcoma patients. More information about this episode and all Osteobites can be found on our YouTube channel, on our website at mibagents.org, and also at your favorite podcast place. And next week on March 16th, we're going to be dropping that fantastic osteo episode. Definitely try to catch that. But we'll be back with Osteobites on March 23rd. We're going to be talking to Dr. Erin Murphy. She's a radiation oncologist at Cleveland Clinic, and she's going to be talking to us about stereotactic radiotherapy, SBRT, and stereotactic radiosurgery, SRS, for sarcoma. So maybe we'll throw out some of those questions to her as well about in response. And you can register for that now. And I think I put the link there in the chat. And then you can find our Osteobites lineup for the next few months on our website. And if you have any ideas for future topics you'd like to hear about, please share them with us at events@mibagents.org. Thank you again to Dr. Bailey and to Vicki and for all of you for spending an hour with us today. And we hope to see you in a couple of weeks when we chat with Dr. Murphy on March 23rd. Thanks so much.